Hello, my lovely anatomists and physiologists. Welcome back. We're ready to finally talk about the process of making new sperm cells, and this is a process called spermatogenesis. So this is production of sperm cells. Now we are going to see that this process is taking place in the seminiferous tubules. So remember, we have here our cut of our scrotum and testis from OpenStack's textbook, and we can see inside the testis, you have just like bundles and bundles of those seminiferous tubules. The other thing to remember is that this is occurring inside the nurse cell. Oops. Which remember we said is releasing some important hormones as well as creating that blood testis barrier. So all of this process that we're getting ready to sort of map out is happening actually inside of the nurse cell. Okay. So we're going to see that this process really begins at puberty. And then, essentially it's continuing throughout the individual's age or throughout their life, although it does sort of slow down as the body gets older, but it's still a continual process. And that's going to be different from what we see with oogenesis in the ovary. We are talking about doing the process of meiosis. So remember, we did have a video on that topic. Make sure that you've picked that up. And so remember, we're going to start out with our stem cell, which is our spermatogonia. And this is going to be a diploid cell. And this cell will go through the process of mitosis, which is more of the same. And so we're just going to basically make a clone. But we're going to call one of those daughter cells of the clone a primary spermatocyte. It's going to be diploid. And then I'm just going to draw this arrow like back up to, it's making kind of a repeat of itself. So one of those cells that's formed during mitosis is going to stay present as a spermatogonia to continue that process. When we look at this primary spermatocyte, we will see that this cell will go through meiosis 1, which is a whole set of prophase 1, metaphase 1, anaphase 1, telophase 1. And we're going to be producing at the end a haploid spermatocyte, and we'll be producing 2 of these. So we're getting at the end of meiosis 1, 2 secondary spermatocytes. Each one of these cells is going to go through a second division. So we're going to see here meiosis 2. So this is a second round of prophase 2, metaphase 2, anaphase 2, telophase 2. And what we produce at the end here will be haploid. We call these cells spermatids. Oops. Spermatids. So it's a haploid cell. And you have 4. And what we'll see if you study meiosis in detail, you'll see that these cells will all be unique from each other. So no 2 spermatid cells are going to look the same. What we see happening next is another process or another step. This process or step we call spermatogenesis. So look at your word carefully. This step here is called spermiogenesis. The entire process is called spermatogenesis. And what we see is this process of spermiogenesis is a maturation process that's taking our spermatid to what we can call a sperm cell. You can call them sperm cells or you can call them spermatozoa. And these are all going to be haploid. But what we're seeing here is that we're going to have our head and then we're going to have our flagella on this cell. And so the sperm cell is really just this flagellated package of DNA material. We see this process of spermiogenesis involves the following steps. So spermiogenesis involves a reduction in the cytoplasm. So when we are transitioning from a spermatid to a sperm, we're reducing our cytoplasm. And we see the maturation into a sperm. And we see that this is stimulated by the hormone ABP. ABP, remember, stands for androgen binding protein. And we see that it's being produced by those nurse cells. So not only is this whole process occurring inside of this nurse cell, but the nurse cell is like promoting this process through the production of the ABP. When our sperm cells eventually leave the seminiferous tubule, they will be considered immobile when they leave seminiferous tubule. And we'll see this process of actually leaving the nurse cell and going into the lumen is called spermiation. So spermiation is the name of the process when the sperm cells leave the nurse cell and go into the middle of the seminiferous tubule. Remember, from the seminiferous tubules, we said they're going to go to the rete testis, and from the rete testis through the efferent ductule, where they head into the epididymis to continue that process of maturation. What we'll see is that the sperm are not mobile until they mix with seminal gland secretions. Remember, we said that the seminal gland secretions are making up about 60% of the total semen or ejaculate. It was high in fructose. So that's the sugar needed for these cells to make the ATP needed to actually propel the flagella. We can say too that the sperm make up about 5% of the total volume of the ejaculate or the semen. So when we did talk about having a vasectomy, which is where you clip, cut, and cauterize the vas deferens, you're still getting a full 95% of the ejaculate. You're just missing the 5%, which is the sperm, which are the cells that can cause pregnancy. Let's go ahead and also say that these cells do not become capable of fertilizing an egg until they have entered the female reproductive tract. So here we're seeing that the sperm have a maturation process that's happening in the testes. We call that spermiogenesis. Then we said there's an additional maturation process that's happening in the epididymis, and we're seeing that these cells can't swim yet. And we talked about there's smooth muscle in both the epididymis and the vas deferens that's helping to propel the sperm actually into the pelvic cavity. We just said that when the sperm mix with the seminal gland secretion, that's when they're actually mobile, but here we see they still can't actually fertilize an egg until they have actually entered the female reproductive tract. So we have this term capacitation that is talking about both mobility and ability to fertilize. So you're here, capacitation is talking about both the ability to swim and the ability to fertilize that egg. Up next, let's take a look at the hormones that are regulating this entire sequence of events, looking at the production of the sperm cells as well as those secondary sex characteristics. So stay tuned. As always, take care of yourselves and each other.